Okay. Okay, folks, so we're going to get kicked off here. I'll just turn up my sound. Okay, folks, so we're going to be kicked off here. I'll just turn up my sound. Okay, folks, so we're going to be kicked off here. Okay, so C18. Okay, folks, so what we're going to cover tonight is basically to give you an idea of what's entailed in the exam, and I suppose how to approach the different types of questions. I'm not going to cover much critical detail. It's more about what's expected in the particular exams. Okay, I'll go through it, and then at the end we'll have a question and answer session where you can ask me about certain topics, about some things you aren't certain about, and we'll take it from there. Does that suit everybody? Yeah? Okay, now I can see you. Is the echoing still occurring at this time?
Doesn't sound like it.
Sounds okay, yeah?
Yeah.
Excellent, okay. So what we're going to do is go through this tutorial and guide you through the exam. Please, if you have any questions, just let me know. Okay. Okay, so I'm going to give you an overview of the exam structure, the criteria of assessment, examples of essay questions in terms of innate essays and adaptive immune response essays, and some examples of short answer questions. Okay, so in terms of the exam, the structure has changed slightly from past papers. The exam itself is still worth 60% of the overall module mark, but the exam is now broken up into 3 sections. And in section A, you'll be asked to complete 12 multiple choice questions, and each of those multiple choice questions will be worth 2.5% each. Okay, so the multiple choice question section contributes to 30% of the exam. Then there's section B, and you'll be required to complete 4 short answer questions, and each of those short answer questions will be worth 5 marks each. And section B will contribute 20% of the exam mark. And section C, you'll be asked to select 2 essay questions from a choice of 5. So each of those essay questions will be worth 25% of the overall exam. Okay, and section C will make up 50% of the exam. Okay, so the overall format, this is how your exam will look. So you'll be as clear as it can be. You'll have 3 hours to do section A, B, and C. And section A will look similar to this, okay? So you'll be given a question like, which of the following cytokines are potent neutrophil chemoattractants? And out of the choice, you'll be asked to circle the the most appropriate answer, okay? So don't forget it's the most appropriate answer. Don't forget that, okay? And as I said, section A is worth 30%. It's compulsory. You have to complete all 12 questions and 2.5% per question. Section B is slightly more challenging. It's 4 short answer questions. Each question will be worth 5 marks, and you should take the time to structure your answer for this to ensure you're hitting key points, okay? So I think the fact that you have 3 hours gives you the opportunity to actually plan and how you're going to provide concise answers in this section. Okay, they're worth 5 marks each, as I said before. And section C, which is where the majority of the marks will be, you'll be asked to select 2 essay questions from a choice of 5. Okay, so this is just an example from a previous exam paper of the type of questions you might come across. Okay, so you'll be asked to select 2 of those essays and complete them within the 3 hours. So the bigger question I think people have is, well, how do I answer the essay questions and where do the marks lie? Generally speaking, when you're asked to answer an essay, what I would expect is that you'll have a structured essay, and I mean structured as in it has some sort of format. And what I'd like you to do is to give me an introduction to the topic being discussed. And we'll have a few examples of how to do this, specifically to complement and are there any questions? And what you'll be asked to do is to provide an introduction to the topic to help the reader get an understanding of the background of the essay. And again, we'll cover critical details of that in the next few slides. Once you've provided your introduction, then get stuck into the meat of the essay, convey the main points clearly, and at this level we'll expect you to provide some evidence of external reading. Okay, so that's where the marks will come from. And lastly, I would expect you to give a conclusion. And when you're giving a conclusion, what I'm looking you to do is to give an overview of how important the immunological process that you've just described is to health. One of the easiest ways to provide an argument to support its role in health is actually to discuss what happens when that immunological process goes wrong. So, for example, to give an example of when defective immune function can result in disease or increase the risk of disease or infection. Okay, so what I'm going to do is give examples of essays from innate immunity in this tutorial, and then I can put up some videos of other examples of adaptive immunity. Okay, so an essay on the complement cascade. So in terms of introduction, what I would like, if for instance complement was to come up, is as an introduction to that essay, I want you to provide me with a summary, a very quick, concise summary overview of what has happened before the initiation of complement. So how did the immune process get to the point where complement activation had occurred? And I'm just going to take you through an example of how you would go through that. Because of course the main thing to be careful here is that the introduction, it needs to be concise. Okay, that's one of the skills to answer an essay, is the ability to present the information in the most concise manner, hitting all the critical points. So be mindful of that. It's not about the number of pages that you fill out, it's how well you articulate the material that you want to convey. So in this example, what I want you to show is that what happens when just before complement's activated. So basically, we're protected from infection by physical and chemical barriers. And what has to happen for us to get infected is the pathogen at hand needs to overcome the physical and chemical barriers to infection. Okay, so that might be stomach acid, it might be mucus, it might be the epithelial barrier, the physical epithelial barrier, it might be lysozyme in the eye, for instance. Okay, so there are some physical and chemical barriers that we have that protect us. And in order for this infectious process to occur, the pathogen has to have found a way to overcome those physical and chemical barriers. And of course, once it's overcome the physical and chemical barriers, the next question is, well, how does the immune system become alert to the fact that we have a pathogen in our system? And the way this occurs is pathogen-associated molecular patterns. So these are components that make up part of the bacteria or virus. So for instance, a PAMP for a bacteria might be part of its cell wall, say for instance LPS, or for a virus it might be genetic material or part of the virus's protein code. Okay, so they're components or antigens, if you like, that are derived from the pathogen. Okay, and these are exposed and released, and the cells in our body express these things called pattern recognition receptors. And what happens is these molecules from bacteria or viruses manage to break off and bind to the receptors on our epithelial cells at the mucosa or our immune cells. And when they bind, it causes the immune system to become activated. So what happens when the PAMPs bind to the pattern recognition receptors on our cells is that it causes our cells to start releasing cytokines. And in releasing cytokines, that's the first step of making our body aware that we have an infection. Because cytokines are essentially communication messages to our body to try to get the right immune cell to the right side of infection to eradicate the infection. Okay, so the pathogens overcame the physical and chemical barriers. The PAMPs that make up the pathogen have managed to bind the pattern recognition receptors on our cells. And when that binding occurs, it causes the release of cytokines. And at that point, the immune system has become aware that we have a foreign pathogen present in our system. And what will happen is the cytokines will activate immune cells within the infected tissue, but they'll also enter the blood to try to alert the immune system on a more systemic level and to attract the right immune cells to the site of infection to deal with the infection. And once these cytokines enter the blood, they'll do a whole range of things, but one of the things they'll do is they'll cause the release of acute phase proteins from the liver. Okay, and these acute phase proteins are broad in nature. You know, you have a whole range of different fibrinogen, CRP, but one of the important acute phase proteins that will be released is C3. Okay, and C3 is critical to complement. So once you get the release of C3, then we will get the arrival of C3 in the tissue, which will then contribute in part to the initiation of complement. Okay, so what I need you to appreciate from that introduction is you have to convey your understanding of the immune response holistically before you start to discuss complement. And the whole point of this process is that in previous years, people explained what complement is, but it becomes quite clear as you progress through the essay, they don't actually appreciate how complement came about and what the reason for complement is in terms of contributing to the immune system. Okay, so in this example, this is how you would introduce an essay for complement. Now, moving on to the main content. Okay, so there's an embedded video in this presentation which will be provided in the on the Blackboard Learn. But in summary, the main points that you need to get across in terms of complement are what makes each pathway different. Okay, there are 3 different complement pathways: the classical, the lectin, and the alternative. Okay, the one criteria that makes that's distinctive about the classical complement pathway is it involves the C1 complex. And the C1 complex is composed of C1Q, C1R, and C1S. The other thing that makes the classical complement pathway distinctive is it's initiated by antibody, and it can also be initiated by CRP, which because CRP can bind to pathogens and basically marking them for activation of the classical complement pathway. What makes the lectin pathway distinctive is instead of having a C1 complex, it has MBL and MASP1 and MASP2. Okay, and that complex will become activated and subsequently cause the degradation, go on to causes degradation of C2 and C4 in a similar way to the classical complement pathway. But the difference in this the only difference between the classical and lectin pathway is that classical starts with C1 complex, whereas the lectin pathway starts with MBL and MASP1, MASP2. Okay, and the thing that kicks off the lectin pathway is mannose components that will be present on the pathogen. Okay. And lastly, the alternative pathway, the thing that makes it different is it doesn't have any C1 complex. It kickstarts due to the random degradation of C3. And when C3 breaks down, it subsequently causes C3b to bind to the microbe, and that kicks off the complement pathway. But the thing that makes the alternative pathway different is it's distinctively different from the C1, from the classical and lectin pathway. The steps in its pathway are critically different. Okay, and the finer details of that will be provided in the video, or if you want at the end, I can give an overview of that pathway. Okay, so you need to know the difference between the pathways, and you also need to know the specific pathway details. Okay, so that's the first part of that essay. The next part you need to be aware of is the whole point of complement is to increase the release of chemoattractants. Okay, and what chemoattractants are components that are chemical mediators that can attract immune cells to the right area of infection. So it enables neutrophils, for instance, to move to an area of low chemotaxin concentration to an area where there's high levels of chemotaxin chemicals present. And the area where you'll have high levels of chemotaxis compounds present is in the infected tissue. Okay, so that's one of the first drivers for the complement pathway, and you need to be able to discuss that. You also need to be able to discuss the rationale for the complement pathway in producing opsonins. Okay, and opsonins are components that attach to the pathogen and enable the pathogen to be marked for degradation. Okay, so opsonins are little molecular tags that bind to the pathogen that enable the pathogen to be recognized by the immune system. Is everyone okay at this stage? Okay, good. Now, let's get back before my computer plays up. Okay, so opsonins are little molecular tags, and in complement, the opsonins generally are C3b and C4b. Okay, they're little molecular tags that attach to the pathogen that enables invading immune cells to recognize that a pathogen is present. It also gives them a little anchor to attach to the pathogen. Again, the finer details of this are in the recorded videos provided. Now, so complement, we've been able to release chemoattractants, and generally they will be C3A and C5A. Okay, and in terms of opsonins, C3b and C4b will behave as opsonins. And the next important thing that takes place in the complement pathway is that they increase the delivery of blood to the site of infection, or that they increase the delivery of leukocytes to the site of infection. And one of the main ways that we increase the rate of immune cell arrival to the site of infection is a very important process called mast cell degranulation. So there are some complement components that are released that will cause mast cell degranulation, and namely they will be C3A and C5A that, when released as a result of complement, will cause mast cell degranulation. And folks, you have to appreciate that mast cell degranulation initiates a massive inflammatory response. Okay, it causes vasodilation, it causes increased vascular wall permeability, and it increases chemotaxis to attract cells to the site of infection. Okay, and it also releases some mediators that will contribute to pain, such as bradykinin. Okay, and there are, in the videos, you'll see the critical detail that's involved in that. My biggest point here is that, please, if you're going to answer this question, if it came up, make sure that you're aware of all the mast cell degranulation mediators, such as prostaglandins, leukotrienes, IL-1, TNF-alpha, IL-8, bradykinin, and so on. Okay, so name them, be able to tell me how they mediate their effects, and lastly, tell me how they result in a symptom. Okay, symptom, for instance, bradykinin stimulates nerve endings, which cause that sensation of pain. Okay, that's just one example. So when you're describing that process, you're talking about what the mediator is, what it does, and how it can result or contribute to a symptom of inflammation. Okay, redness, swelling, pain, whatever it may be. Okay, so what will then happen is all the leukocytes will infiltrate the tissue, and they will subsequently attach to the opsonin that's attached to the pathogen, and they will phagocytose the pathogen. Okay, but there's one last step that needs to be talked about, and that is the formation of the membrane attack complex. So the formation of a membrane attack complex is the only direct way that complement kills a pathogen. Okay, complement kills a pathogen through a lot of indirect ways, i.e. through the recruitment of leukocytes, but actually the membrane attack complex is a way that complement can directly kill the pathogen. Because what happens is you get C5b as well as C5b, C6, C7, C8, and C9 forming a pore in the pathogen, and when that pore forms, it causes the osmotic lysis of the pathogen. Okay, so that's complement. Now, the other thing to consider is once you've done the the main content. The next thing you need to discuss is the importance of complement in health. And one way to discuss that is to relate how defective complement can enhance the risk or severity of disease. Okay? And this is where your reading outside the notes come in. It is present in the notes, but you can also read around it. And it's talking about what happens in some individuals that have defective C3, or what happens in some individuals when they have defective ability to form a membrane attack complex. Okay? And you would give an overview of the fact that these deficiencies can happen and how they can impact the risk of disease or the risk of infection. Okay, so once again, introduction, main content, and lastly, a context or conclusion. How does this immunological process impact health? Okay, now, is everybody okay with that so far? If anybody has any questions, just let me know. No. Okay. Moving on to phagocytosis. Again, it's just another example of how you would structure your essay. Okay, here we are again, the same format, introduction, main content, conclusion. So, the introduction for this particular essay would be slightly different, okay? Because it's near the end of the process. So you have to be a bit more concise in how you describe how we got to that point in the immune response. So again, you're talking about the pathogen has overcome the physical and chemical barriers. Its PAMPs have bound the pattern recognition receptors on our immune cells, or our mucosal cells. That's caused release of cytokines, which has caused the initiation of complement, leading to opsonization of the pathogen and increased arrival of immune cells to the site of infection due to the increased blood flow as a consequence of leukotriene and prostaglandin release, increased chemoattraction of the right blood cell to the site of infection due to C5a and IL-8 release. And lastly, it's easier for the immune cells to arrive at the site of infection due to the fact that the blood cells have increased their permeability due to IL-1 TNF-alpha. Okay, so at that point, what we've talked about is the pathogen has infected the body, caused initiation of the immune response due to PAMPs binding PRRs, and what happened after that was complement increased the blood flow to the site of infection, and chemoattractants ensured that the right blood cell got to the site of infection, and they've made their way into the infected tissue where they are about to phagocytose the pathogen. And now we then go on to talk about the process of phagocytosis. Okay, so folks, you've got to remember you're trying to tell a story. Okay, and in this case, you're given a synoptic overview of what happened in order to get us to the point where the neutrophil is going to phagocytose a pathogen. Is everybody clear on that? Has anybody got any questions on that? It's a difficult task, but if you want the high marks, that's what you're going to have to be able to do. Sorry? Okay, we're still good to go, yeah? Excellent. Okay.
Yep.
Right. So, we've talked about the main content, okay? Or sorry, we've talked about the introduction. Again, it's that very short story about how we got to this point of phagocytosis. And the next thing we need to do, and again, this is an embedded video which you can watch when it's provided on BB Learn. But essentially, what you're going to talk about is the fact that the pathogen has been opsonized. Now, the reason it's important to talk about the fact that the pathogen is opsonized is if the pathogen is not opsonized, it makes it difficult for the infiltrating neutrophil to anchor onto or attach to the pathogen. So the opsonin that's bound to the pathogen could either be C3b or C4b, or it could also be antibody. Okay, so it could be antibody bound to the pathogen or C3b or C4b from complement. Now, how does the neutrophil bind to these components that are bound to the microbe? It uses specific receptors. Okay, so neutrophil will have Fc receptors, and these receptors on the neutrophil enable it to attach to the Fc region of antibody. And by attaching to the Fc region of the antibody, it enables the latch on and subsequently phagocytose, or take up the microbe and the antibody to break it down. Likewise, if it's C3b or C4b, the neutrophil will use its CR1 receptor. Okay, and that will enable it to attach and phagocytose the material. So once it's attached to the pathogen, yeah, patience, yeah, absolutely. Two ways to do it. Obviously, from an innate point of view, it's going to be C3b, C4b. And from an adaptive immune response, it's going to be antibody. Does that make sense? Oh. Okay. Now we're worried. So once it's attached, then what we have to do is to encapsulate it and take it into the cell. And what happens is you get vigorous extension of these pseudopods, these almost like arm projections, wrapping around the pathogen and taking it into the cell. Now, when it's encapsulated and takes it into a cell, it's referred to as an endosome, as a phagosome. Okay, and it's in the phagosome. But the challenge we have now is that in order for the phagocytosis to occur appropriately, we need to acidify the phagosome to enable the enzymes to work at their optimal pH. And the way we achieve this is that the phagosome will fuse with an endosome in order to acidify the phagosome. Okay? And the way it acidifies it is the endosome will provide proton pumps to allow for acidification of the phagosome. Okay, as you can see here, the late phagosome will be a pH of 4.5. Okay, so now we have an acidified phagosome. The big event that has to occur is that this late phagosome has to fuse with a lysosome. And when it fuses with a lysosome, it forms a phagolysosome. Okay, and an important thing to note is that a molecule present on the phagosome called EEA1 enables the phagosome to be directed towards the lysosome to allow for this fusion. Okay, at the end of that process, we have the phagolysosome. Okay, so that's a summary of that part of the phagocytosis. Now what you need to discuss is, well, okay, we've got the formation of the phagolysosome. How does that result in the destruction of the pathogen? It results in the destruction of the pathogen through a whole range of processes, but the two main processes that you'll want to discuss are the respiratory burst and enzymatic mediated degradation. So in terms of respiratory burst, it's oxygen mediated degradation. And the concept of the respiratory burst is to generate radical species, that is reactive oxygen species, that will degrade that will react with the proteins, the DNA, and the lipids of the pathogen, causing the structures to break down. And of course, if the structures, such as the protein present in the microbes, if they break down, then the microbe is essentially becomes dysfunctional and essentially will be lysed. Okay, so you have to appreciate that the respiratory burst degrades the pathogens through the generation of radical species, which will degrade structures that are essential to survival for that pathogen. Okay, and you'll need to know about NADPH oxidase and the enzymatic reaction that takes place, as outlined here. Also, then you'll need to know about superoxide dismutase and lastly myeloperoxidase. And appreciate that the formation of a hexafluorous ion, OCl-, is a very critical radical species that is very effective at degrading the pathogen. And also just to appreciate that you can have the formation of a nitrosyl radical species as directed here. Okay, so they're the radical species that will contribute to the degradation of the pathogen. And the other thing you have to appreciate is this whole process takes place in this encapsulated phagolysosome. And the important thing to note is that because it takes place in this encapsulated phagolysosome, it protects the neutrophil itself from the radical species. If the radical species were to get released in the cell, it would cause it would create a potential danger to our own tissue. And sometimes in some conditions, the respiratory burst can get so out of control due to excessive infection that in fact it will cause tissue damage. Okay, but in this case, because it's occurring in a phagolysosome, it's in a restricted environment, which means damage is limited. So that's the respiratory burst. Another thing that takes place is enzymatic degradation. And these are, generally speaking, oxygen independent. And you need to talk about things like lactoferrins, which essentially sequester iron from the cell. And one of the reasons they will sequester iron is in order for the pathogen to survive, they need iron availability. So if the body has a way to sequester iron, then it makes it more difficult for the pathogen to thrive. You also get the release of lysozyme, which will degrade the peptidoglycan present in the bacterial cell wall, for instance. You'll have the release of defensins, which have the ability to form pores in the bacteria membrane. And then you have a whole catalog of enzymes that will break down the pathogen structural components. So glycolysases will break down sugar components, phospholipids will break down membrane components, and nucleases will break down the genetic material. Okay, so that's a quick overview of phagocytosis, the main content. So the next thing is to provide a conclusion. And one way to provide a conclusion or context is to discuss what happens when you have defects in phagocytes or phagocytosis. Okay, things like chronic granulomatosis disease, or where you'll get defective respiratory burst due to defect NADPH oxidase, and also you can have type 1 leukocyte adhesion deficiency LAD1. Okay, so look for other examples in terms of extended reading, and that way you can convey a good understanding of phagocytosis and its importance. Okay, so is everybody happy at that stage?
Yes. Okay.
Okay. Okay, so now we'll just give a quick overview of potential adaptive essays. Okay, here we are again, the same structure, introduction, main content, conclusion. So in terms of somatic recombination in B cells, so questions in this particular context are based around the fact that from the adaptive response, we need to develop a broad antibody repertoire to enable us to respond against a broad range of pathogens, okay? So this is the whole concept that, you know, we need each B cell has its own unique B cell receptor that is specific to a pathogen. Therefore, we need a broad range of different B cells with different B cell receptors that will respond against the different pathogens. Now, considering the vast amount of pathogens that we have the ability to encounter, it means that we need to have a broad range of B cell receptors possible to deal with that. So, what you need to convey in the introduction for this particular type of essay, where you're going to talk about how we achieve B cell receptor diversity, what you need to convey is why we need to achieve B cell receptor diversity. And what you're going to say is the innate immune response is useful, but there's a need for it to be assisted by the adaptive response, because the innate response is not specific and does not enable us to respond to develop an immune response specifically against a targeted pathogen. Okay, so when we have an innate response, it's just a general, for instance, if we have a bacterial infection, the innate response will be a general antibacterial immune response. Whereas in the adaptive response, what happens is instead of having a general antibacterial response, it has a response that's specific to that bacteria. So for instance, if we have salmonella infection, then our adaptive response will develop an immune response not against bacteria, but specifically against salmonella. Okay. Now, what's the question here? Can you still hear me? Do we still have visuals? No? Right, okay. Okay, I see it's frozen there. Yeah. Okay, I will try to stop the sharing, then reconnect. Are we good again? Good, okay. Okay, so we'll try to get started again. Excellent. Okay. Right. So what we want to do at this point is provide a rationale for why we need a specific response. So, as I said before, the innate response is useful for a general, quick, rapid, non-specific response, but there's a real need to have a tailored response against that single pathogen that we've been infected with. So in order to develop a specific response, we need unique receptors in our B cells and our T cells, okay? Now, are we all aware that each B and T cell in your body has its own unique receptor, and that each B and T cell in your body is designed specifically to target only one pathogen, okay? If I took a B cell from my body now, it would only be able to respond to a designated pathogen that its B cell receptor can recognize and bind to. Are we happy with that? Are we aware of that? Okay. Now, one thing that you need to be aware of is that essentially what, okay, nice. Yes, Joanna, we need to know it depends. We need to know the difference between naive cells and immature cells. Okay? Immature cells, it's a different answer, but I'll cover that in a minute. Whereas naive cells might all have different B cell receptors. The only thing that makes them naive is they haven't encountered antigen yet. A copy will be provided on BB Learn, yeah. Okay, so what we need to be aware of is the fact that, alluding to your question, Joanna, when B cells are immature, that is before they develop, every B cell in your body produced that's immature all have the same copy of the B cell receptor genes. Okay? So they all have the same copy of the B cell receptor gene. And essentially what happens is each one of those B cells undergoes a process of somatic recombination and hypermutation that results in them changing their B cell receptor genes in their own unique different way. Okay? So every B cell changes their B cell receptor genes during this developmental process in a way that they do it, every B cell does it differently. And that's a good thing, because if every B cell does this somatic recombination process differently, it means that every B cell will have a different version of that B cell receptor gene, and as a result, they'll have a different B cell receptor. And if they have a different B cell receptor, then it means they'll be able to each B cell will be able to recognize different pathogens. Does that make sense? Okay. So development of unique receptors is achieved through somatic recombination and junctional diversity through somatic hypermutation. And this will allow a B cell receptor or T cell receptor to be developed specifically for antigens with that are unique to a particular pathogen. And this will enable our immune response to develop a response that will specifically target a given pathogen. Okay. Now, there will be videos provided, as I said before, but these are the main things that you need to cover in this essay. You need to cover heavy chain rearrangement. Okay, so that's the V, the D, and the J rearrangement. And don't worry, folks, there will be detailed videos provided. And what I'll do is I'll provide it, and over the coming weeks, I'm open to answer any questions in regard to the content. So you talk about heavy chain rearrangement in terms of V, D, and J. Once you've done that, you'll then talk about light chain rearrangement. Once it undergoes heavy chain rearrangement, it'll be tested using a surrogate light chain. And once that's successful, it'll then progress to light chain rearrangement. And once it undergoes light chain rearrangement, it then will progress and become a mature B cell. But some of the details that you'll have to discuss are things like allelic exclusion and junctional diversity. You also need to include the mathematical calculations or the mathematical details that show you how much diversity is created through somatic recombination and how junctional diversity contributes to increasing that number. You'll then also be required to talk about how, when a B cell receptor becomes activated, it will move into the follicle where it will manipulate its B cell receptor a further time to create a highly specific B cell receptor against the pathogen. Okay. So there's again an attached video. So what happens when the B cell receptor becomes activated? It enters the follicle. It stops expressing its B cell receptor. And the reason it does that is, at this point, the B cell has produced a B cell receptor that recognizes a particular pathogen. But at this point, once we've been infected with a pathogen, at this point we want to improve the affinity of the B cell receptor for that pathogen. Okay, so at this point it is able to recognize the pathogen, but we want to fine-tune the B cell receptor to give it even better affinity for the pathogen. So once the B cell becomes activated, it moves into the follicle, stops expressing its B cell receptor, and it does this because at that point that will enable the B cell, or the centroblast as it's referred to at this stage, it will enable it to manipulate the B cell receptor gene through the removal of nucleotides between the V, D, and J segments and between the V and J segments of the light chain. And by manipulating the B cell receptor gene components, it will alter the affinity of the B cell receptor for the pathogen. Now, that will happen in one of two ways. Some of the cells or centroblasts that manipulate their genes will increase the affinity of the B cell receptor for the pathogen, and in some of them it will reduce the affinity of the B cell receptor for the pathogen. And that's fine, because what then happens is those cells that manipulated their B cell receptor to increase affinity for the pathogen will be selected and given a signal to survive. And the way it does that is those that have a higher affinity for the B cell receptor, or for those that have a B cell receptor, those that have changed their B cell receptor in a way that increases the affinity for the pathogen, will be able to interact with the follicular dendritic cell. And when that interaction happens, those cells get a signal to live. Those cells that have changed their B cell receptor gene in a way that has less affinity won't be able to compete to bind to the B cell receptor, or won't be able to compete to bind to the follicular dendritic cell, and as a result they will not get the signal and they will die. And inevitably what happens through this whole process is you get the production of B cells that have manipulated their B cell receptor to produce a high affinity B cell receptor for the pathogen. Okay. Once it's then done that, it will interact with the follicular helper cell to class switch. And what I mean by that is every time a B cell receptor becomes activated for the first time, it will always express IgM. But at this stage, the follicular helper cell will instruct the B cell to either stay as IgM or to change to IgA, IgG, or IgE. And the reason it will instruct it to change is that it will enable the B cell to produce an antibody that is best designed for the site of infection. For instance, if we have an infection in the gut, it makes more sense to have an IgA antibody. Okay? If it's a parasitic worm, then it makes more sense to have an IgE antibody. Okay? And that's the principle of class switching. Once it's class switched, it then either becomes a plasma cell or a memory cell. And at that point, the plasma cell will produce massive amounts of antibody against the pathogen, whereas if it differentiates into a memory cell, it's there for subsequent future infections. And at that point, if we get reinfected in the future, the memory cell can kick off this process very, very quickly. Anybody, any questions? Now, there's better animations in the recordings that will be provided. But that's a quick synoptic overview. Does anybody any questions? Does everybody understand the process? Or do they feel they need maybe read up a little bit more on it? Okay, that's perfect. That's completely understandable. Good, excellent. Well, hopefully I'll be able to provide you with a recording that will make this process a little bit easier. Okay, that's fine. Judith, what instructs the change from IgM, etc., is cytokines released by the follicular helper cell. Okay. Is that clear, Judith? Yeah? Okay, cool. Okay. So don't forget, folks, tonight is about trying to, I suppose, educate you on what's required in the essays, as opposed to the general detail of essays rather than critical detail, which you can pick up from the videos. So again, just to highlight, the main content is give an overview of heavy chain rearrangement, talk about the fact that it needs to be tested against surrogate light chain, give an overview of light chain rearrangement, including the processes of allylic exclusion and junctional diversity. Then give an overview of the mathematical probability details to explain the number of possible combinations, and then talk about that after B cell activation, or when the B cell encounters antigen, it moves to the follicle, where it undergoes another process of B cell receptor gene adjustment. Okay. Now, in terms of context and conclusion, again, we're back to this process of, okay, we create a diverse range of antibodies or B cell receptors. How is that important? You could allude to what happens when we don't have sufficient B cell production or B cell receptor production. Okay, and at this stage, in terms of context conclusion, you would relate to some of the immunodeficiencies that exist. Okay, and discuss how how defective B cell maturation or defective class switching can contribute to serious health issues. Okay, so at this point, it's taking this content and mixing it with some of the content from the later lectures in immunodeficiency. Now, is everybody happy with that? And not the details, but maybe more, I'm just looking to see if you're happy with what's required. Good. Okay. Now, moving on to adaptive response to a virus. Okay. So one of the things that's critical to an immune response to a virus is antigen presentation. Okay. And in terms of, you're going to talk about antigen, the endogenous antigen presentation pathway, in terms of the introduction, I need you to tell me what happened before antigen presentation. So how did the immune system know that a virus was present? And at that point, what you're going to talk about is the virus overcame the physical and chemical properties, and the PAMPs from the virus bound to pattern recognition receptors. And it would be useful if you could include examples of viral antigen or viral pattern recognition receptors, such as RIG and TLRs. Okay. And what happened then was the viral components that bound to the PAMPs caused cytokine release. And in causing cytokine release, it caused type 1 interferon and type 2 interferon to be released. And you could discuss how interferons contribute to antiviral activity. For instance, decreasing gene expression in surrounding cells and inducing cell death of infected cells. Okay. And another mechanism in terms of the innate response to a virus is the activation of natural killer cells. Okay. And the fact that natural killer cells check for expression of MHC1 on cells, and if cells don't express high enough levels of MHC1, the natural killer cell will kill it, because the natural killer cells essentially deems that if there's low expression of MHC1, that a virus must be inhibiting MHC1 expression. Okay, so you basically just give an overview of how the body responds to a viral infection from an innate point of view, that is interferon release and natural killer cell activity. And then you're going to go on to the fact and say, like, you go, okay, so an innate response is useful, but there's a real need for a tailored response against the specific virus that has infected the body. Okay. And one way that we achieve that is through activation of the adaptive response. And actually what kicks off the adaptive response is the antigen presenting cell. And in this case, it's generally the dendritic cell that will collect an antigen and bring it to the lymph nodes. Okay? But also the infected cell itself will express MHC1 in terms of antigen presentation. Okay? And in terms of antigen presentation, the main content is present in the video. Okay, it's a whole process of the virus infecting the cell, making its own proteins, the proteins getting broken down by the immunoproteasome, and the epitopes released from the broken down protein, moving to the endoplasmic reticulum through TAP, and then subsequently through the use of tapicin and the chaperones in MHC1, the epitope of the pathogen will get loaded onto MHC1 and delivered to the surface. But again, the main content of that will be present in a video provided on BBLearn. Okay, in terms of context and conclusion, you would give an example of how a virus is able to stop the antigen presentation pathway. And here in this slide, you can see a few examples how, for instance, Epstein-Barr virus can inhibit the immunoproteasome, and there are a whole range of other viruses that will inhibit the TAP, that will inhibit the formation of MHC1, or that will inhibit the arrival of MHC1 to the surface of the cell. Okay, so again, in the terms of the context, you're saying antigen presentation of a virus is very important, but this is what can happen, or this is how viruses are able to override this protective presentation pathway. Everybody happy with that? Okay. Now, okay, and lastly, what I'm going to cover is the type of introduction and context that you would provide for a question that focuses on naive T cell differentiation. So this is the differentiation of a naive T cell into either Th1, 2, 17, or Treg. Okay. What you would do is you would talk about, again, this whole idea of how the immune cell became activated, causes the release of cytokines, which cause the activation of the innate response. But whilst that innate response was undergoing, then the adaptive response also got kick-started. And it kick-started by the fact that dendritic cells collected antigen from the site of infection and migrated towards its local lymph node. When it migrated towards its local lymph node, it directly went to the paracortex of the local lymph node, where it presented antigen to the naive T cell. Then you would talk about how the T cell became activated, signal 1, signal 2, and the fact that the naive T cell then differentiates into either Th1 or Th2. So that would be introduction. Then you would have your main content, again, which is present in the videos. And in terms of context, you would start to discuss what happens when T cell differentiation becomes defective. Okay, for instance, T cell differentiation or the CD4 T cell differentiation can become defective in things like HIV infection or through other genetic defects. Okay, and the other thing you have to appreciate is what this essay is relating to is the differentiation of a helper cell. Okay, and the thing to appreciate is that helper cells are required for all adaptive immune responses. Doesn't matter whether you need a CD8 response or a B cell response. Both of those responses require help, and help comes in the form of CD4 T helper cells. For instance, a Th1 response is needed in order to activate CD8 T cells if it's a viral infection. A Th2 response is required to respond against extracellular pathogens such as bacteria, and they will provide help to B cells. Okay? But what I need you to grasp is that yes, we have the introduction, then we have the main content of what happens when the T cell becomes activated. And lastly, I need you to provide an overview or context of what happens when you get defects in CD4 T helper cells. Okay, folks, that's me for now. I'm now going to take some questions. I would prefer, you won't lose marks for using acronyms, but you'll gain marks by fully expressing the acronyms. Absolutely, Joanna. No problem. Patience, that's the question that it's not about the amount of pages that you write, it's about the content, okay? Now, if you consider you have 3 hours to do this exam, so you have plenty of time to put considerable content in there. Yes, Judith, it will. Faye, the coursework marks I have them, but they need to be moderated because there are some issues with plagiarism, so I have to finalize those, and then I would definitely hope to have your marks back before the 3-week deadline, which is the 22nd of December, because we have to provide coursework within 3 weeks. Yeah, 40%, yeah. So you have to get 40% in both the coursework and the exam. Good luck, Judith. I wouldn't bet on it. if I'm being honest. Philomena, I would inquire about that and get the feedback. It should be there. Judith, you will not have an issue with time. 3 hours is buckets. If you consider the amount of time it will take to answer 12 MCQs and 4 short answer questions, you'll easily have that done in the first hour, I would imagine, which gives you an hour each for 2 different essays. Just take your time, Judith, to plan it. On the day also, I promise you'll have sufficient time. Take the time to structure your essay. Just write it out in little skeleton form, and then subsequently go about it. Burnage basically, right? You'll have a certain space provided. Provide as much detail as you can. I don't know how to make that more clear. If you look, there is a document on BB Learn. Yeah, Eleanor, yeah. There are some examples on BB Learn. I'm guessing that's where you see that, Eleanor, yeah? Okay, perfect. Negative marking, thankfully, is not a thing. No problems, Judith. Good luck with it. I don't envy you. No problem, Eleanor. Hold on. Burnage, in terms of grammar and spelling, is your main thing is to be able to get across the critical points. Although I would expect it, there are limits to what I can allow to pass. I need it to be legible. That's the main thing. The other side. Cozy, you have limited space on the sheet, but if you really wanted, you could provide more detail in other sheets that will be provided.
Thank you.
No problem. No problem, Patience. Good to hear. Cheers, Bernich. No problem, Pat. No problem, Jessica. You'll get there. Enjoy Christmas, then get stuck in. Absolutely. Folks, just to remind you is to complete the module valuation survey, please. Cool. Bye. No problem. Have a good one. Yes, Donna. Generally speaking, that's how it'll happen, yeah. Same to you, Philomena. I think there was a few problems, Donna, unfortunately. The answer to what, Donna? Hold on, I go up. Oh yeah, I see your question. I don't know. Yeah, generally speaking, yeah, it'll be on the test. The section 1 and 2 will be on the test paper, and then section 3 will be on in provided answer booklets.